So according to Chargaff's rule, number of purines will be always equal to the number of pyrimidines in the case of the double strand DNA. Reason, we have already learned whenever there is purine on the one strand, it will be pyrimidine on the opposite. And if it is pyrimidine on the one strand, it will be purine on the opposite. So the numbers are going to be equal. That means A plus G, because purines are which one? A and G. Pyrimidines are which one? T and C. So the number of A plus G is equal to number of T plus C. So you can say A plus G ratio T plus C is always equal to 1. And but remember one thing, when you talk about A plus T and G plus C, because we know number of A will be equal to number of T. Reason, A will always face T and G will always face C. So the number of A plus G is always equal to T plus C. But A plus T will not be equal to G plus C, because these are going to be varying from species to species. Now the amount of A plus T divided by G plus C is the ratio, and this ratio will be low in the case of primitive organisms. But when we talk about advanced organisms like human beings, amount of A plus T is more compared to G plus C. There is a very easy reason behind it. A plus T develops only 2 hydrogen bonds. G plus C develops the 3 hydrogen bonds. In the primitive organisms, amount of G plus C was more because it develops more hydrogen bond, and more hydrogen bond means more stability. And we all know in the primitive time when evolution started, conditions were unfavorable. So the organisms had to survive in the unfavorable conditions. So more stability was given by more amount of G plus C, 3 hydrogen bond. As the evolution continued, the amount of A plus T increased because conditions have become more favorable for life. That's why more amount of double bonds have come, because there is already quite sufficient good conditions. So the number of bonds came down to A plus T. A plus T carries 2 hydrogen bonds, G plus C carries 3 hydrogen bonds. Let's understand this whole concept with the help of the diagram. So this is how the bonds get formed. If you consider this as one nucleotide, because see, it's a complete diagram, you can say it's a pentose sugar. Don't worry about OH and H and all, because we are simply learning the diagram of nucleotide. So this is one nucleotide, pentose sugar, or you can say deoxyribose sugar. Then the first carbon sugar is going to develop a N-glycosidic bond with the nitrogen base. First position in the case of purines, and first position in the case of pyrimidine, and 9th position in the case of purines. Now the next thing is, the 5th position carbon will develop a bond with the phosphate group, and this bond is called as phosphoester bond. Now when one nucleotide comes in contact with the next nucleotide, then develops a bond which is called as phosphodiester bond. So chain formation requires phosphodiester bonds. Now what is phosphodiester? Let's understand. This is one nucleotide up to this. So the second nucleotide will develop a bond with the first nucleotide with the help of its phosphate. If you remember, this point I had made it clear in the beginning also to you, that phosphate has 3 hydroxyl groups around it, and that's why the phosphate is able to develop more bonds, because these are reactive groups. They can easily perform bond formation and release a molecule of water. So what exactly happen is, the phosphate of this nucleotide will develop a bond with the OH present here. Remember, this sugar carries an OH at the 3rd position. So this OH will participate in bond formation with the other OH. So finally what will happen? A molecule of water is gone, and now this phosphate has developed a bond with the previous sugar, or you can say with the 3rd carbon of the previous sugar. So the phosphate participates in 2 bonds, you can see here. one bond is with the previous sugar, with the third carbon, second bond with the next sugar, and that is with the fifth carbon. So phosphate develops two bonds. That's why these bonds together are called as phosphodiester bond. This is how a complete chain will be formed. Now you can see in this, this carbon carries a OH, it's reactive. Now this OH can develop a bond with the next phosphate, then next phosphate, it continues like this. So a chain of alternating phosphate sugar, phosphate sugar, phosphate sugar, this will continue. So you can see in this picture now 5 prime end. Now pay attention to one more thing. One end is 5 prime end, the other end is 3 prime end. So why do you call it as 5 prime? Because here reactive group is still present. Fifth position phosphate group is free, and in this case third position component is free. That's why 5 prime end and 3 prime end. So every chain starts with a phosphate. From its head will always have a phosphate, and tail will always have the sugar. So phosphate is present on the fifth position, then sugar, then phosphate, then sugar. You may continue like this. So now remember, first phosphate will participate in phosphoester bond, but rest all phosphates will participate in phosphodiester bond. Please be clear with this. So the first nucleotide phosphate group will be attached with the phosphoester bond. Rest all phosphates are attached with two bonds on both sides. Now this is what I was talking about on the last sheet, that 5 prime end to 3 prime end, 5 prime end to 3 prime end, anti-parallel to each other. Second point we learned on the last sheet was that nitrogen bases are towards the inside. So you can see it is towards the inside of this diagram. And whenever there is A present here, opposite will be T. Whenever there is G present here, opposite will be C. And two hydrogen bonds and 3 hydrogen bonds will be there.